discussions, the first of the lectures of clinical chemistry. So our topic today is acute pancreatitis, but it's not like a medical topic, but we will discuss it as a clinical chemistry. So acute pancreatitis, it's actually the inflammation of the pancreas, but just we go direct to the enzymes that is important in the case of the acute pancreatitis, which is the first one is the lipase. The most sensitive test of acute pancreatitis is the lipase, because if it's the concentrations of the lipase 3 times more than the normal one, it's one of the criteria for the diagnosis of acute pancreatitis. So the most sensitive one is lipase. Okay, and increase its level between 2 to the 50 times than the normal. So if it's more than 3 times more than the normal, then that is the diagnosis here is acute pancreatitis. So it will be in increases between 2 to the 50 times than the normal, and then it increases in the first 4 to the 8 hours and reaches its peak at the 24 hours. So it will be in increases in the first 4 to the 8 hours and then reaches at the peak at the 24 hours. So it will be after 1 day, or at 1 day, it will be the largest concentrations of the lipase enzyme. So it will be in increases in the first 4 to the 8 hours till it reaches its maximum at the 24 hour. So now we talk about the other enzyme, which is known as amylase enzyme. So if we look at the amylase enzyme, the molecular weight, it's about 57.7 kilodaltons. So it's 57.7 kilodalton. It's a protein actually, and it's composed of the 511 amino acids, and the tubular reabsorption is 50% occur, so it appears normally in the urine. One just important thing to make a comparison between the lipase enzyme and the amylase enzyme, that the lipase, it's impossible to find lipase in the urine sample, but the amylase enzyme, except for maybe the um, abnormal conditions, but the lipase normally it not present at the urine at all. But the amylase, 50%, it occurs 50% of it reabsorbed in the tubular or through tubular. So that means it 50% of it will return to the blood, and 50% it will appears in the urine. So it normally appears in the urine in the case of the amylase enzyme. And then chymotrypsin, chymotrypsin appears in the feces. So the chymotrypsin appears also in the feces, and we have also the chymotrypsinogen. So amylase, 50% of it appears in the urine, so normally it appears in the urine, and chymotrypsin appears in the feces. Chymotrypsinogen synthesized and stored in the pancreas. Single polypeptide chain of 245 amino acid. I'm just curious about if the chymotrypsinogen it's synthesized in the pancreas, even it's a protein, because most of the protein synthesized in the liver, but this protein chymotrypsinogen is synthesized by the pancreas and is stored also in the pancreas, and it's composed of the single polypeptide chain of 245 amino acids. So it's a large number of amino acid cross-linked by 5 disulfide points. Okay, so it will be a strong protein. So the chymotrypsinogen is synthesized and stored in the pancreas, single polypeptide chain composed of many 245 amino acid, and they are just cross-linked by the disulfide bond. It composed of the 5 disulfide bonds. And here we have chymotrypsinogen by the trypsin, then that is lead to the chymotrypsin. Okay, by the trypsin that is can be happen, because we have here chymotrypsin and chymotrypsinogen. And maybe the question that comes to your mind, what is the relation between the chymotrypsin and the chymotrypsinogen? Actually, the enzyme that is responsible for that is a trypsin, because a trypsin, it's a break down these disulfide bonds, or cleaves disulfide, cleaves 15 unit of the polypeptide chain from N-terminal, and that is happen in the small intestine. That will be turned to the chymotrypsin. That's why we see it in the feces, because if you just understand this equation, you can understand why we see the chymotrypsin in the feces. Actually, we're talking about the normal condition. So we have here chymotrypsinogen by the trypsin that is present in the small intestine. Everything that is happening in the small intestine turn into the chymotrypsin. How that is can be happen? It cleaves 15 units of the polypeptide chain, and that is happening from the N-terminal. That is happening in the small intestine, so the chymotrypsin appear in the feces. And there is something is called the trypsinogen. The trypsinogen, it's self-activation by cleavage of a hexapeptide after secretion into the small intestine. No need of the enzyme. So the trypsinogen, there is no need enzyme for it. It just self activation by it just, you do everything by itself, like do the cleavage of hexapeptide, and that is lead to the self activation after the secretion of it into the small intestine. Once the trypsinogen see itself in the small intestine, then that is happen self activation, and that is lead to the cleavage of the hexapeptide. Determined by the immunoassays are a proteolytic enzyme that hydrolyze the same substrate. So it acts as a proteolytic enzyme and causes hydrolysis of the substrate, which is called trypsinogen. Zymogen, it's inactive precursor of the enzyme, so it's in active form. By cleavage of one or more covalent bond, transforms into the active enzyme. And then we go to the glucose. It's one of the most common preformed analytic methods. So the glucose is one of the most important analytic method. It can be measured in the blood, in the plasma. It can be measured in the urine. And you remember, if we measure it in the plasma, we use fluoride, FLCL. We use the fluoride chloride. And we measure also the glucose in the urine. We measure the glucose also in the cerebrospinal fluid. So it's one of the most common performed analytic method, and we measure it in the several things, like in the blood, the plasma, the urine, and also we can measure it in the cerebrospinal fluid. So we have the glucose concentration in the blood, which is 10 to the 20% less than the glucose concentration in the plasma. Acid here in the plasma, which means that the concentrations of the glucose in the plasma more than in the blood. Why? Because the glycolysis occurs in the whole blood tube. Okay, so which leads to decrease the glucose concentrations. The question is here, why the glucose level in the plasma is 10 to the 20% more than the glucose that is present in the whole blood? Because the glycolysis, which is a breaking down of the glucose, that it's happened when there is a whole blood. I don't know why, but if there is a whole blood supply, then the glycolysis that is happen in this situation. Okay, and that is lead to decrease the glucose level. That explains why the glucose level is 10 to the 20% in the plasma more than the glucose that is present in the whole blood. Now we talk about the reference interval for the healthy individuals. We said before in the milligram per deciliters, and we said also in the millimole per liters. And as you can see here, it's 70 to the 100. Actually, it's the best thing that we can see in the glucose level. If we just said, oh, okay, 74 to the 106, it doesn't matter, okay? But actually the range, it's between 70 to the 100 milligram per deciliter. In the case of the millimole, it's actually between 4.1 to the 5.9 millimole per liter. And then we discussed also what is increasing the glucose level, like diabetes, Cushing, because of increasing the cortisol, stress also increasing the cortisol, so that all of them increasing the glucose. Insulin is anabolic hormone, yeah, right, but it decreases the glucose level. Starvation decreases the glucose level. Adrenal insufficiency also, it decreases the glucose level. So we have many methods of measurement of the glucose. We can use hexokinase method, we can use glucose oxidase method, and actually we have also the glucose dehydrogenase enzyme. So we have many levels or many methods to measure the glucose level, as we can see here. So the first one, I will start with the hexokinase method. So our enzyme is hexokinase, and from its name it just add phosphatic group to our glucose. So here we have the glucose, okay, and we add this enzyme which is known as hexokinase, and as you can see here, it's reversible. Okay, in the hexokinase it will take phosphatic group. Do you think from where to get phosphatic group? From ATP. So hexokinase take the phosphatic group from the ATP and add it to the glucose. With which part? To the 6 carbon. So it will be glucose 6 phosphate plus ADP. Deal, deal. Then the glucose 6 phosphate, we do the another enzyme is called glucose 6 phosphate dehydrogenase, and dehydrogenase, that's mean the results, it will be NAD from to the NAD positive, and that is lead to the 6 phosphogluconate. So we can measure it by this way. So another the first method is hexokinase method. We make the glucose plus ATP by the help of hexokinase. It will be turned to the glucose 6 phosphate plus ATP. Then the glucose 6 phosphate by the enzyme glucose 6 phosphate dehydrogenase, that is used NAD to the NADH, that is lead to the production of 6 phosphogluconate. But the question is how this reaction help us to measure the concentration of the glucose. Just focus that the glucose 6 phosphate dehydrogenase, that is lead to the 6 phosphogluconate. That's not the deal, but the deal is here, because we know the absorbance for the NAD on NADH positive is 340 nanometers. So in this reaction we can measure the absorbance of this reaction, and if it's 340 nanometers, then we know exactly that the glucose 6 phosphate or here is a glucose. Okay, the other method is glucose oxidase and method. Okay, so the first one, hexokinase, what is the calcification of the enzyme? Is transferase, so it's calcification 2. Now we go to the oxidase calcification 1. Good. So we have the glucose, oxygen, and H2O, water, by the glucose oxidase, that is lead to the gluconic acid plus H2O2. Then the H2O2 plus O ansinidine, which is colorless compound, by the peroxidase enzyme, that is lead to the O-andesine plus H2O. Now we just explain more about this reaction. So we have the glucose plus O2 plus water, so we need oxygen and we need water, and then we use the enzyme oxidase. Glucose oxidase, that the problem is not for the gluconic acid, but the problem is not the problem, actually the solution, or we can say the focus here, that is lead to the production of the H2O2. And then the H2O2 with the colorless compound, whatever this colorless compound, by the help by the peroxidase enzyme, that is results to the colorful compound plus H2O2. So we can measure the glucose by this mechanism. So the first one, hexokinase method, we said glucose ATP hexokinase lead to the production glucose 6 phosphate plus H2O. Plus ATP, and then the glucose 6 phosphate by the glucose 6 phosphate dehydrogenase, NAD, that lead to production NADH, and that is lead to the 6 phosphogluconate. All the focus is to the NAD to the NADH, because we know the absorbance is 340. Then the other method is glucose, or we can say glucose oxidase method. It's just to turn it to the H2O2, and then the H2O2 with the colorless compounds by the help of the peroxidase enzyme that will turn to the colorful substance plus H2O. Then we know that there is a glucose here. So we use two enzymes actually. One method is hexokinase, the other method is glucose oxidase enzyme. Then we go to the glucose dehydrogenase enzyme. Glucose by the NAD positive to the NADH by the glucose dehydrogenase enzyme that is lead to the gluconate dehydrogenase. What is the calcification? 1 exactly in the oxoreductase. So we have the glucose by the help of glucose dehydrogenase enzyme that is lead to the gluconate. We are not looking for the results actually, we're just looking to this reaction that the glucose dehydrogenase enzyme use NADH to the NADH positive. So we measure the NADH production measured by the photometric at the 340 nanometer. Okay, and he give here is another substance like NADH plus colorless compound by the dihorse or whatever that is lead to the colorful substance, but actually the best and the most common that we measure it by the photometry, which is 340. What is the criteria for the fasting glucose level? The fasting glucose level, if we just tell the patient please fast, and then we just measure the glucose, just like don't eat, don't drink from the 12 o'clock or something, because we just make the patient fasting for 8 hours and then come in the morning and we measure it. And if it's more than 126 grams or milligrams per deciliter, or more than or equals 7 milligram per deciliter fasting glucose level, if it's more than or equals 126 milligram per deciliter, or more than or equals 7 millimole per liter, then that is diabetes. Okay, clinical medical decision, clinical medicine decision limit. It's the name clinical medical decision, or we can say clinical decision limit, and we have discussed that before, that we said that this limit or this reference that is important for the medical guidelines for the treatment, for the urgent treatment. So when I said fasting glucose level more than or equals 126 milligram per deciliter, or more than or equals 7 millimole per liter, they have the same value. Okay, that means this patient diabetic and we should deal with the patient as soon as possible. Okay, then we go to the ketone bodies. This is the third time that we discuss the ketone bodies, and I have told you before that the ketone bodies it explained in another file completely and in details, but we just focus on some points. When we just repeated it, we just focus on some points. So when we have excess acetyl coenzyme A, acetyl coenzyme A, that is important for the production of the energy as we know, but if we have the excess of it, that is lead to the production of ketone bodies. Like this patient, the question is how this patient or how this human has excess acetyl coenzyme A, because he has excess intake of the fat more than the carbohydrate, or because he is diabetic patient, or there is a rare metabolic disease that is lead to the excess amount of the acetyl coenzyme A. So we have many mechanisms of that. Maybe this man take too much fats, excess intake of the fats, or he is diabetic patient, or something else, that he has a rare metabolic disease, and that is lead to the excess of the acetyl coenzyme A, which lead to the production of the ketone bodies. Now we go to other things here, which is known as the beta cells. What is how the productions of these hormones? So the productions of the several hormones like glycogen, somatostatin, and insulin, all these cells, they are produced from the islet cells of the pancreas, which is called the islet cells of the Langerhans. As you can see here, the beta cells is in the center of the islet cells of the Langerhans, which is responsible for the production of the insulin. And here we have the alpha cells and the delta cells. The alpha cells produces glycogen, and then the delta cells produces the somatostatin. So we have, as you can see here, the islet cells of the Langerhans. In the center, beta cells responsible for the productions of the insulin, and then we have the delta cells for the somatostatin, alpha cells for the glycogen. What are the functions of the insulin? And I think there is something wrong here, because uh, actually the insulin is anabolic hormone, as you can see. It's responsible for the glucose influx. That's mean it reduces the glucose level in the blood, so it maybe turn the glucose to the glycogen, so it reduces the glucose level. Pyruvate to the glucose actually against that, it's actually reduce the glucose level. Acetyl coenzyme A to the fatty acid, it's anabolic actually product protein, and it's against the lipolysis. Actually it doesn't cause increasing the level of the fatty acid. Okay, it's responsible for the protein synthesis, gene expression, amino acid uptake, potassium sodium pump, DNA synthesis, apoptosis, and also autophagy. So I think that is against autophagy and against apoptosis, because insulin is anabolic hormone. So we have two types of the diabetes. Type 1 diabetes, that is present in 5 to 10% of all cases of the diabetes, 75% before the age of 18 years, and abrupt onset of the symptoms, like the patient, he doesn't know that he has diabetes, he's so young, 18 years, 17 years, 16 years, in that age, and he just come with the polyuria, polydipsia, and decrease weight and decreasing of the weight, or like weight loss. So that, like when I said abrupt onset of the symptoms, like he didn't know at all that he maybe have a diabetes, like a polyuria, maybe said to himself that maybe I just drink so much water or something, but he will has polyuria, polydipsia, and also weight loss. Autoimmune process destructions of the beta cells, so that is lead to the insulinpenia. Destruction, it's autoimmune process, and that is lead to the destructions of the beta cells. When I said autoimmune process, because the body attack itself, so that is lead to the destructions of the beta cells, and that is lead to the reduce the amount of the insulin of the body. For long term, that is lead to the ketosis. So this patient should take insulin forever to prevent the ketosis. Then we go to the type 2 diabetes, which is more common. It's present in 90% of the cases of the diabetes. Initial minimal symptoms, not born to the ketosis. Actually, it's actually not lead to the ketosis. Impaired insulin action, it actually has insulin, has beta cells, but impaired in the insulin action. Maybe the insulin doesn't act so well, or the receptors doesn't respond to the insulin. Therapy include the first diet interventions, change lifestyle, and then the insulin, and then the drug. It's actually drug and then the insulin, the final treatment. Mostly onset, it's when you just the patient older or more 40 years old, but these are, we can say, the typical symptoms when I say type 1 diabetes and type 2 diabetes. Then we go to the glycated hemoglobin A1C, and we know that is like most important thing for the diagnosis of the diabetes. It's not enzymatic. Again, it's not enzymatic. Addition of the sugar residues to the amino acids of a protein. Actually, it's a protein, and we add the sugar residues to it, and this process is called glycation. Okay, not glycosylation. So we have to be to be careful about our pronunciation. It happens by glycation, because the glycation when we just make sure, okay, the glycation or glycolization, both of them add sugar residues on the amino acid, whatever, if I'm just talking about the hemoglobin A1C or I'm talking about the another proteins. But the difference here is the glycation, it's not enzymatic, but the glycolization, it's enzymatic. And we said many times that this reaction or addition of the sugar residues to this amino acid, it's actually not enzymatic. So we can say it's a glycation of the amino acid. That's mean addition of the sugar with no help of the enzyme. Hemoglobin A1C is by a glycation of N-terminal valine. So valine is one amino acid that is present in N-terminal residue of each beta chain of hemoglobin A, because we know that the hemoglobin composed of two alpha subunit and two beta subunit. Okay, and what is happening in the case of hemoglobin A1C, it just we add a sugar group to the N-terminal of this amino acid, especially the valine. So we add this sugar, and we said again it's a glycation, not a glycolization of the beta chain of hemoglobin A1C. So the glycation happened on the beta chain, not on the alpha chain. So as you can see the N-terminal amino group, and we add just the alpha chain, and we have here the hemoglobin A. Why we said hemoglobin A? Because it's for the adult, and we add this glucose level, so that is lead to the albumin, which is a pre-hemoglobin AC, and then it's happened amide rearrangement, then this lead to the hemoglobin A1C. As you can see here that the hemoglobin A and the glucose, it will be rapid reaction to form the pre-hemoglobin A1C, and then it will become slow to form hemoglobin A1C. So again, hemoglobin A, it started with the hemoglobin of the adults, and then we add a glucose. As you can see here, we didn't mention any enzyme, so we can say it's a glycation. Okay, and it's reversible, as you can see here, and it's very rapid, and that is lead to the pre-hemoglobin A1C, and by the slow reaction that is lead to the hemoglobin A1C. It's similar to the glycolization of extracellular matrix protein. It's similar to what is happened to the glycolizations of the extracellular proteins. Methods for determination of the glycated hemoglobin. So we have many methods how to describe or how to detect the glycated hemoglobin, like hemoglobin A1C. It's actually more than 150 methods to detect that. One of these famous methods, like a charge paste method, that it's depend on the charge and positive or negative, or the structure paste method on the structures of this hemoglobin A1C, or enzymatic and optical methods. So we have a large number of the methods that we can use. So the first one is a charge paste method, like ion exchange chromatography, or high performance liquid chromatography, or capillary electrophoresis, or isoelectric focusing. And I have just mentioned them. I'm just not talking about the details of each one. All of them, they are a charge paste method. The first one is ion exchange chromatography. The other one is high performance liquid chromatography, and then the capillary electrophoresis isoelectric focusing. Then we go to the structure paste method, affinity chromatography, and immunoassay. So we can use ELISA method, which is immunoassay, and we can say it's immunoabsorbent assay. And we use affinity chromatography that focus on the structures of the it. The other thing, we can use enzymatic method, photometry, spectrophotometry, or mass spectrometry. Then we go to something is called lattice turbidity, which is measuring of the light scattering as a lattice particles, how they are just scattering the light. They just coat it with a specific antibodies and react with the hemoglobin A1C in the samples. And after these antibodies react with the hemoglobin A1C, that is lead to the aggregations of lattice particles, because they just forming a complex between them, between the hemoglobin A1C and the antibodies. So that is lead to the aggregations of the lattice particles, and that is lead to the change of the turbidity. And that can be quantified. The steps: the first one, we just give the blood sample preparations, what we call hemolysis, because we need hemoglobin. So RBCs after the hemolysis, that is lead to the productions of hemoglobin. Then we make an incubation with the lattice particles, like we put the hemoglobin with these lattice particles. And I said before that the incubation, we just make this on appropriate conditions. As you can see here, that there is a hemolyzed RBCs, and we see this is a free hemoglobin, and then we put the lattice particles. The lattice particles added to the hemolyzed blood. The lattice particles have high affinity, non-specific to all hemoglobin forms, whatever, if it's hemoglobin not glycated or hemoglobin A1C. So after we add lattice particles, I'm sure I'm talking about the lattice turbidity. As you can see here is hemolyzed blood, so we will have a free hemoglobin, we have also glycated hemoglobin, so we have different types of the hemoglobin. The lattice particles has high affinity for every single hemoglobin, so it will bind to all of them. And then we add the addition of the anti-hemoglobin A1C, which is monoclonal antibody, and we just said before that the monoclonal antibodies are more specific. So we add anti-hemoglobin A1C monoclonal antibodies. We just add antibodies against the hemoglobin A1C, which they are non-mouse anti-human hemoglobin A1C monoclonal immunoglobulin antibodies is added. We can say it's a reagent too. So this is what we call them as antibodies, mouse anti-human antibodies, and this is the hemoglobin A1C, so they are bind together, and that is lead to the formation of the lattice antibody complex. And after we add this, what should we do? We addition of the good anti-mouse antibodies. It's like the same mechanism that what is happening in the Western blotting, okay? The same thing. We just add another antibodies, addition of the good anti-mouse antibodies, but it's polyclonal antibodies. And after we add, this is the glycated hemoglobin. This is the mouse anti-human immunoglobulins or antibodies, which they are monoclonal antibodies. And then we add the mouse goat anti-mouse antibodies. So that is lead to the agglutination. They are just making agglutination at the bottom of the sample, and then we see these clumps. So the quantity of the hemoglobin A1C, that is proportional to the degree of the agglutination, proportional to the turbidity. So they will be just form clumps at the bottom of the sample that we can see it, and in increasing these clumps, that's mean in increasing the concentrations of hemoglobin A1C, in increasing the turbidity. Now we go to the glycolizations of the proteins, which is different to what is happening to hemoglobin A1C. We have a carbohydrate which is glycan, and then it's form a covalent bond with the protein. So we have two types, N-glycolizations and also O-glycolizations, and that is depend which terminal that the glycolization is happen. Actually the N-glycation that is happen in hemoglobin A1C, because we have amino acid valine and the glucose is attached to the N terminal of valine. So we will start in N-glycolization is attachment of the sugar to the protein. So as you can see here, it's N, and then it binds to the, for example, an amide of the asparagine, because we know that the asparagine, it has side chain of the amide group, but actually to the N of the amide group. Okay, and that is lead to the N-acetyl glucosamine. Okay, N-acetyl beta D glucosamine. Or glycolization, attachment of the carbohydrate to the hydroxyl group of the amino acid, and the glycan is attached to the serine, threonine, hydroxypurine, hydroxylysine, which is so common in the serine and the threonine that they have OH. So they bind to the serine, to the threonine, so we can say it's O glycolization because it bind to the hydroxyl group. So that is lead to formation of an acetyl alpha D-galactosamine. So in the case of N-terminal, that is lead to the production of an acetyl beta D-glucosamine, and here an acetyl alpha D-galactosamine. Many glycoproteins contains multiple sites when the sugar can be attached, lead to the heterogeneous mixtures of glycoprotein forms. So many glycoproteins contains many sites when the sugar can be attached, lead to heterogeneous mixtures of glycoprotein. The sugar chain can attach to the multiple sites of the protein, so that means the glycoproteins it has many isoforms. Then the glycoforms microheterogeneity, as you can see here, the same protein, but it attached to the different sugar chains. Glycolizations doesn't directly encoded by the DNA of the protein, so it's varied in the factors that is affected it, like it's not coded directly by the DNA of the protein. The availability of the specific sugar molecules, activity of the glycosyl transferases, cellular location, environmental conditions. So we have many factors that is affected where like there is not a code, like there is no DNA that is affected the glycolization. So many factors affected it, like the availability of specific sugar molecules, activity of glycosyl transferases, cellular location, environmental conditions. It's actually the research topic now. And then the glycolization is a species specific, tissue specific, cell specific, protein specific. It's synthesized actually in the endoplasmic reticulum. So we have a sugar group by the help of the enzyme that is lead to the glycoprotein, and all these happen in the endoplasmic reticulum. Antibodies are glycoprotein, the sugar part identified as identifier. The sugar part we here said a paratope of the antibodies, and that is make the immune system to recognize it. It's antigenic determinant, and we can say it's ABO system. As you can see here, it's so complicated that we have the P and acetyl. We have the first thing is beta and acetyl galactosamine to 1,3-P galactose, beta and acetyl galactosamine, and then that lead to the non-reducing form alpha L-fructose type A blood group, and then the beta galactose that is lead to the alpha L-fructose, and that is lead to the type P blood groups. So the glycolization is easily impaired by the regular ethanol intake. If these patients take too much alcohol, so the glycolization it will be impaired. Carbohydrate deficient transferrin is used as a sensitive marker for alcohol abuse. Yes, another thing that is showing us that this patient is alcohol abuse, that we use a test called CHO, which means carbohydrate deficient transferrin. So the transferrin is a transport protein that carries iron in the circulation. It's a transport for the iron. Each transferrin can carry 2 atoms of the iron. There's something is called total iron binding capacity, which is determined by the transferrin. Does the transferrin can carry more iron or not? Number of the open sites on the transferrin could bind additional iron are referred as unsaturated iron binding capacity. It's unsaturated. If it's unsaturated, that means it can carry more iron. So the total iron binding capacity or unsaturated are measured in the diminished lab. And then we have isoelectric focusing, as I just explained. That is one of the measuring of the glucose level, as you can see here. And we have the congenital disease disorders of glycosylation. So we have type 1 and we have type 2. So the type 1 is the early steps of the glycosylation, while type 2 is the late steps. It's incomplete or incorrect additions of the sugars in the type 1. The enzyme that added the sugars or under the glycosylation, so the sugars it will be decreased. That is lead to developmental delay, liver problem, seizures, muscle weakens, affect the initial attachment of the sugar in the proteins, and where it's the location endoplasmic reticulum. But in the case of the congenital disease of glycosylation, it's in the late steps. It's a proper processing or modification of the sugars. So enzyme that is modified, the effect is in the enzyme that is modified the sugars. Improper process wrong substructures of the sugar. The same thing, developmental delay, liver problem, seizures, but plus immune system and muscle issues. It affects the final modifications of the sugars on the protein, so everything that is happening in the Golgi apparatus. As you can see here, that all the discussions about the first lecture. Thank you so much for listening.